I'm going to be doing some of the neurodevelopment part of your developmental biology course. This year, I don't know if anyone. I know that Auntie was there for the last year. And last year, what I did was try and do vertebrate neurodevelopment and invertebrate neurodevelopment together. But this time, luckily, what we've done is we've divided up the course. So your next 4 lectures are going to be neurodevelopment, and we've divided. There she is. Oh no, sorry, no. So what we've done is we've divided up into invertebrate neurodevelopment. So you'll have 2 lectures of that. Your next 2 lectures will be that. And then 2 other lectures, which are vertebrate neurodevelopment. And Bhavna is going to take those 2 lectures. So 4 lectures, 2 on invertebrate, 2 on vertebrate. I will do the invertebrate and Bhavna will do the vertebrate, but we'll both, as much as possible, try and be there for all 4 lectures so that we can keep this interactive and go back and forth between vertebrate and invertebrate development. Okay, so I put my email address up there. So if you have any burning questions on developmental neurobiology, developmental biology, evolution, anything, please do shoot me an email. Usually I would have said accost me in the corridors or walking around in the canteen. Unfortunately, that doesn't happen so much anymore. But if you do see me, do stop, and I'm always happy to talk talk development. Okay, so let's get started. I don't know, is Bhavna, are you here? Not yet. Okay, but she said that she might join a little bit late and will leave a little bit early, but I think she will join. Okay, so I like to. Oh, it's not moving ahead. Hello. Okay, I like to start with this picture, which represents the tree of life. And when I first saw this, I was quite astonished because in my head, I had imagined the tree of life to be plants and animals and nothing more than that. And then when I first encountered trees of life that looked like this, I was amazed at all of the different phyla that I had no idea of. And on this figure, what you can see, I don't know. Yeah, okay. On this figure, what you can see are these yellow circles. I don't know if you can see them with my pointer. The yellow circles indicate where there's multicellularity in this tree of life. And all the black circles indicate that there isn't any multicellularity. And so the reason I show this is because the first thing to notice is that there are very few instances where you even have multicellularity, right? To consider where a nervous system could be. And even in this spotty multicellularity that you see in this tree of life, nervous systems are only present in this one tiny little twig of the tree of life. And so really something special to consider, right? So we're studying an organ system which is not present in a vast majority of organisms on Earth. Now, what does the nervous system allow you to do? So here's a video of Bhushan the cat. Now, what the nervous system allows an organism such as Bhushan to do is to perceive the environment through sight, sound, taste, smell, and then decide to execute a response and bring together all of the structures of itself to execute that response. And that response has to be really quick because whatever it's attacking cannot get away. In this case, it's my finger, so it should be able to get away. But usually it needs to be really precise to make sure that that prey doesn't get away. And so the nervous system, so first of all, animals, the animal kingdom, has the ability to respond to the environment in exquisite ways with exquisite complexity. And what allows them to do that is complex nervous system that allows it. So the nervous system is allowing Bhushan to sense all of these environments through all of these cues and integrate it, decide what it wants to do with all of those cues, and then after the decision is made, let all of the organ systems know this is what you need to be doing, coordinate it all, execute that response with precision, accuracy, and speed. right? So not nervous systems that allow this complex behavioral repertoire in organisms have necessarily got to be complex. Okay, so since I started off by talking about that tree of life, let's consider nervous systems as they evolved. So there are many different kinds of nervous systems. And the earliest nervous systems were not that complex. They were what people refer to as diffuse systems. So here's that tree of life, and now what we're doing is zooming in to just that animal part of it. So here is the base of the tree of life. So your fungi over there, and the rest of all of this are the animals that we were considering. And if you look at the animals that diverged early, so that's your sponges, your placozoa, all of this business, they don't have nervous systems. Nervous systems tend to have, you begin to see them over here. So your ctenophores, your hydrozoas, your jellyfish, all of these begin to get things that you could call nervous systems. And if you look at that, so here's an example of from this little branch here, the hydrozoa. If you look at the nervous system these organisms have, they're like a network of cells within the body wall that are connected to each other and have the other properties of nervous systems. That is, they can respond to the environment, evoke a calcium response, and talk to each other through it, and allow the organism to execute a behavioral response. So you can see that in this beautiful movie made not too long ago, where these guys looked at a hydra responding to the environment, and they were monitoring calcium activity in the neurons. And you can see the simple nervous system allows the organism to respond to behaviors. But it's not a complex nervous system. Complexity started arising when through evolution, these networks of neurons began to get condensed into, maybe, in these, you may have seen gastropods. Where are the gastropods? Here we go. Over there, you may have seen and dissected. I don't know if this is done anymore in bachelor's and masters in zoology and things, but you may have seen ganglia, dissected out ganglia, looked at condensations of neurons that form these processing centers that are called ganglia. And then further complexity arises because these ganglia come together into something much more complex, which you can call a brain. So, okay, just a quick point. I I can't see anyone, so I don't know. I don't know if I'm getting across or not. And so I would like everyone to stop and ask questions if you don't understand something, if I'm not making sense, or even if you just want to crack a joke, I'd be very happy. Just so that I know that there are people behind those names over there. And I'll tell you that I have only 22 slides. So I've made it such that we can really talk about these things and not get inundated with just information. I'd like it to be interactive. So please do stop and ask, and we'll learn about nervous system development that way. Okay, so complexity arose by bringing all of these things together in a centralized way. And so now let's look at a complex brain. Oops, how do I proceed without it playing? Okay, so here now what we've done is the same thing. So these authors, so here's a larval zebrafish, and the blue line over here, why is it not? Oh, damn it. Okay, doesn't matter. But the blue line over there is showing you the outline of the body wall of the larval zebrafish, and you can see that the brain is condensed. And what they're doing is they're monitoring neuronal activity, just like you saw in the previous slide. But now you can see there are much more neurons, and the pattern of activity is very, very it's complex, right? There's all kinds of things happening, different structures coming up, lighting up, and then suddenly this network of activity in the entire structure. So this is how complexity arose. So what we're going to talk about is, how do you get that through development? Okay, so let's talk a little bit about that complexity. People have been trying to understand this complexity. So the first way that you would do this is to look at the cells, to open up the brain and see what it looks like. So you may all have seen pictures at your local zoo of these Golgi stained brains, all kinds of organisms, from turtles, from pigeons, flies, human tissues, mouse, and he made these beautiful pictures of neurons connecting with each other. This picture that I'm showing you is from Jeff Lichtman's lab, and it came out maybe 5-7 years ago. So this is the new way of labeling neurons and looking at them in brains. This was probably the first iterations of these. It's called Brainbow. And what you're doing is taking a genetic label and randomly allowing it to be expressed in all of the neurons. What you're looking at is a tiny fraction of the mouse hippocampus.
Sonia,
Yes.
I think someone had a question. Yojit had a
Yes.
question, I think.
Yes, please do direct me if there is a question. Who had a question?
Yeah. So someone raised their hand.
Please,
Yojit,
come on, ask away. Yojit, okay.
Hi Sonia. I just wanted to ask, evolutionary speaking, what role does what purpose does it serve for Hydra to have these neural networks?
The purpose of the nervous system is essentially something that even single cell organisms can do, right? You need to perceive the environment and mount an appropriate response. What's happened in the Hydra, as you become multicellular, you can't rely on the systems of, say, signaling gradients that allow you to respond to stimuli. So then you need to come up with something that allows you to sense the environment and execute a response. The Hydra is using the first instance where it's sort of segregating, saying, okay, I've become probably too multicellular to rely on those simple signaling gradients and things that single cell organisms use. I need to dedicate a tissue for this. And the first solution is you produce your sensor all across the body, so that maybe you could sense a noxious stimulus at one end and tell the other end of your body that there was a noxious stimulus over there. So evolutionarily, the nervous system in the Hydra is doing the exact same thing that a single cell organism would do, but it's having to do it in a different way because it's multicellular now. Multicellular beyond being able to do it using the methods that single cell organisms could use. Does that make sense?
Yes.
And the thing with evolutionary questions is it's always conjecture. You can never really say why. You can guess and you can try and test those guesses, but you can never really pin it down. More and more you can, but they're hard questions, evolutionary questions. Did anyone else have questions? Okay, so then let's get back to this Brainbow image of the vertebrate, of the mouse hippocampus. So the reason I'm showing you this is if you look at, sorry, my screen is split, so sometimes it may look weird. Okay, so can you see my pointer pointing to the cell layers of this image? Yes. So this is a densely packed. So the reason I'm showing you this is look at the number of neurons packed into that tiny volume, and look then at all of the processes that it's sending out, right? And this is just a tiny fraction of a tiny part of the mouse brain. And actually, people have tried to map out connectivity, and they've done this. They've taken an example in the mouse brain of about a 1 millimeter cube of the mouse brain, the cortical part of the mouse brain, and they've tried to reconstruct every single neuron, every single synapse within that volume. And they make an estimate of even that there are about 100,000 neurons in that volume. And again, this is an estimate with about 1 billion synapses. That's the level of complexity of nervous systems that we're talking about. The reason I'm talking about this is because I want you to keep thinking, this is a developmental biology course. So I want you to keep thinking that how do you get the genome to make this, right? How do you get the genome to make 100,000 neurons for this small volume? And how do you get them to make those 1 billion connections? How could the genome encode for something like that? This is the mouse brain, but think now what happens when you're considering the human brain, right? The numbers are so vast that we get poetic about this. So here, so I've got all of this. You'll see when Varsha uploads this, I've put in the notes where I got these numbers from. So it's a paper, it's a Nature article from a few years ago. So please do have a look at this. But really, this is, I think, a quote directly from there. So see, you know, in humans, estimated to have 100 billion neurons. I don't know what that means. My brain can't process that. And looks like the authors can't either, because they say roughly the same number of neurons as there are stars in the Milky Way. Okay, and then all of those neurons have a 10 to the power 15 connections. So that's the scale of complexity we're talking about. And remember, we want to ask ourselves, how could the genome encode all of this complexity? Any guesses? Do people know how many genes in the human genome?
Around 30,000.
Around 30,000, exactly. And look at what that genome has to do. Okay. So people have tried to do this to simplify this problem, right? It's too big. Milky Way is too big to handle. So you look for simpler organisms. Have you guys encountered C. elegans in any of your developmental biology lectures so far?
Not yet.
Yes, no? Shubha, what did you
Not
say?
yet.
Not
Not
yet.
yet.
Okay. So a hot favorite developmental biology bug that's used, model organism, is C. elegans. And we've learned a lot from C. elegans. And in the 80s, Sidney Brenner, who you may have heard of, along with his colleague Benjamin White, actually decided that they would map all of the cells of C. elegans, and there aren't that many of them. And one aspect of that was mapping every single neuron. So because the C. elegans is such a simple organism, they could actually go in, count exactly the number of neurons that made, so this is not estimates now, exactly the number of neurons that make up the C. elegans brain. And they said it was about 300. And they could count, and this is through electron microscopy. And because it's electron microscopy, you can go in and count the number of synapses, the exact number of synapses. So this was in 1986. And actually, I want you to look at, can you see that ring-like structure? I don't know if it will be clear on many of your screens, but there's a red ring-like structure over there. That's the brain of C. elegans. So very simple. It's a ring with nerve cord traveling all the way to the posterior of the organism. And then there have been refined ways of doing this with fancier ways of fixing tissues for EM that preserves tissue better. And that came out about last year. And they basically, so now what we know in C. elegans is an EM reconstruction of all of the neurons of the hermaphrodite, the male, and the male C. elegans nervous system. And what you can then do is map, once you know exactly which neurons are, and you've done EM, you can map the connectivity, which neurons connected to what. And that's what you can see over here. All of this is connectivity. I can see Ayanti, you have a question?
Yes, I was wondering, so from 302 neurons and the numbers of synapses, has there, do we know how many, so I suppose this number of synapses is how many connections, whatever circuit junctions there are. So I'm wondering if there is like a back of the envelope kind of calculation that one can do to say one neuron connects to how many others.
This
or
is it. It's not, Auntie, it's not back of the envelope. This is it. This is exactly what they have done. And usually, I do not remember now exactly if this diagram does this, but what it's basically doing is telling you that connectivity. So all of these, I mean, it looks like a mess, but do go look at this. It's telling you exactly those connections. And sometimes when it's not at this resolution, they'll pick up a few and they'll represent the numbers. So this neuron is connected to this one by so many synapses, whereas this one is connected to the other one by much fewer. So in something as simple as the C. elegans, you can actually map that very, very precisely, and they do that.
Okay.
But similar efforts, you can imagine, have been taken in more complex organisms. So here is Siona intestinalis. Does anyone know what that is? It's called a sea squirt. Does anyone know what a sea squirt is? Okay, if you don't, then you have, then when this class is done, go look it up. And go look up a joke about tenured professors and sea squirts. I won't tell it here. But so Siona intestinalis, or the sea squirt, is one of the earliest vertebrates. Its larval form has this nervous system, and its adult form is actually sessile. It doesn't need a nervous system. So it kind of looks like a sponge in the adult form. And if any of you have been to any places where ships are docked or little harbors, and you look on if the water is clear enough, you can look, there'll be these sponge-like things that stick out. Those are sea squirts. Anyway, so the larval form has this nervous system, and that is, again, just about 200. So very simple vertebrate, very simple nervous system. And that, again, this group has mapped exactly like in the C. elegans. And it has, unlike the C. elegans, it has, given the number of neurons, it has more synapses per neurons. Also, of course, these two are very simple. There's a ring. This one kind of has a brain, but these are very simple to be able to really understand complexity, right? And so then this critter over here is kind of sits at a sweet spot between this simplicity, which doesn't really not satisfying from a developmental biology perspective, to the complexity that I showed you in the mouse brain. So this brain kind of sits right in the middle of that spectrum of complexity. So this is the fly brain. It consists of about 200,000 neurons, if you were to count all of the neurons in the fly, and about 100 million connections. So this is kind of getting in the realm where you don't really know how a genome could build such a thing. And the other thing about the fly is that it displays a huge variety of complex behaviors. So flies have exquisite ability to fly and navigate. They have exquisite ability to smell and see. They learn. They show circadian behavior. They show aggression. They demonstrate very elaborate courtship rituals. So fairly complex brain, which executes fairly complex behaviors. So the fly kind of sits in that sweet spot where you can begin to ask questions about how does the genome encode complexity of an organ like the brain. And then, of course, you guys have heard about flies, so you know what phenomenal genetics you can do with the flies, right? You can fiddle around with genes at specific times and specific cells. You can put in genes, you can take out genes, you can do that with multiple genes at the same time. So now imagine this organism is sitting in a sweet spot of complexity of behavior and nervous systems and is genetically tractable. Now you can answer questions about how do you build this complex nervous system. Okay, so we're going to talk about this. The first thing that we're going to talk about, and actually we'll spend most of our time on this, is how do you generate all of those neurons to begin with, right? Because you have in flies. Okay, anyone knows how many genes in the fly? Any guesses? So humans, we said 30,000. In flies?
900?
A little bit more than that. Who's what do they do at auction? They have that, what is that called? Going one, going twice. What's it called, Yojit?
Bidding.
Yeah. So does anyone have another bid?
13,000.
13,000, that's getting close. Yeah, so it depends on how you count, whether you count microRNAs, genes, non-coding, all of that. Somewhere between 13 and 17,000 is the number of genes in the flies. And how many neurons? You got 200,000, right? So we need to use that genome to generate all of this diversity. And we're going to spend most of our time talking about how you could do that. The next question it will come up in your mind as you see a structure that's at the back of that slide over there. All of those connections, how on earth, even if you make these 200,000 neurons, how on earth do you get them to make the right connections? Because imagine if you make the wrong connection, that is a fly that is doomed to die, right? It will not survive. So how do you ensure that that genome makes the right connectivity? And then, of course, because anytime you're working with development, the structure makes sense in light of what it's supposed to do. So even if you're working in development, you can't help but think about how does that circuit then produce behavior, right? And then, of course, this is what I've tried to do from the beginning of the talk. You always want to keep thinking, how did this evolve? Because when you keep that question at the back of your mind, how do you construct it becomes a much easier thing to understand if you look at other critters. So very important when you're doing developmental biology. We study organ systems using tractable organisms for as model organisms in developmental biology. But remember, they represent a very tiny fraction of biology on Earth, and so it's very useful to just keep your eyes peeled and look at other little branches on that tree of life. See what you can learn from there. Okay, so but primarily in these talks, in the next 4 talks, we will talk about how do you generate all of those diversity of neuron types, and we may touch upon how does it assemble into circuitry somewhat, and then of course we have been talking a little bit, just keeping our eyes peeled about, evolution. Okay, so here's the overview of all 4 lectures. Like I said, we're going to split it into vertebrates and invertebrates. Can you guys hear me clearly? Because there's some construction outside, and I want to be sure that you don't have.
Yes.
You can. Okay, good. Okay, so so we're gonna talk vertebrates and invertebrate development, and then we're gonna break it down, right? And what you'll notice in both Bhavna's and my work and my talk, that there are some common themes. So you'll notice that whether you're looking in vertebrates or invertebrates, you have to do to build a nervous system, you have to go through it in a stepwise process, and that's what I'm showing you over here. And then the other thing you'll notice is that while the steps may be, there's similarities, there are also differences. Keep your eyes peeled for those, and also keep your eyes peeled for the mechanisms that allow you to get there. There are very interesting similarities and very interesting differences, both of which tell you about important things about development of the nervous system. So the first step, whether you're looking at vertebrates or invertebrates, is in that developing embryo, you need to set aside a tissue to say that this is going to be what gives me neurons. So this is neurogenic. That tissue is neurogenic. You need to set aside neurogenic tissue during embryonic development. In invertebrates, this is called the neuroectoderm because the nervous system is an ectodermally derived organ. So it's the neuroectoderm in invertebrates. In vertebrates, you call it the neural plate. Same idea. You set aside tissue and give it neurogenic potential. So you will see that. We'll talk about how do you do that. And then there's slightly different things happen in vertebrates and invertebrates. In invertebrates, now you take that tissue and then you say not all of you will be neurons. There'll be some subsets of you which will retain the potential to make neurons. You don't want everything to become a neuron. So you do that in invertebrates. In vertebrates, you'll see Bhavna will tell you how you take a neural plate and fold it into a tube, and that's your starting material for making the rest of the nervous system. And so there's some interesting similarities and differences over there. Then once you have this tissue, this neural tube, or the progenitors that have delaminated, we'll come to that in vertebrates. Now you need to give it positional information, because remember, the nervous system is developing all the way from here to, you can't see, but all the way down to the bottom, right? So you need to tell the nervous system that this part is going to be anterior, whereas something over here is going to be posterior. Over here, you need to make the brain. Over here, you need to make the spinal cord. So you need to give that tissue positional information. So this is called spatial patterning. There may be slightly different terms used. In invertebrates, we talk about spatial patterning. I think in vertebrates, you call it regionalization or specification of regions. Same idea, you're giving positional information. And when we're talking about positional information, there are two axes of positional information. You'll be familiar with this from your early developmental classes in Drosophila. You need to specify the dorsoventral axis, so ventral dorsal axis, and you need to specify the anterior posterior axis, like I said. So you'll see how this is done in both vertebrates and invertebrates. And then, of course, we will talk about something that we've already kind of referred to, which is cell division. So very basic cell biological concept of how do you get the numbers of neurons that you need. So cell division, different types of cell division play into this, and we'll talk a little bit about that. And then finally, we'll talk about temporal patterning, and we'll save that for later. Okay, so let's take this one by one. So let's first talk about specifying the neurectoderm. So any questions? Okay, so we're now in the business of taking that Drosophila embryo that you guys have built in Tina's sections, and now you want to set aside that tissue that will retain neurogenic potential, the neuroectoderm. So can someone, okay, I'll just put that down there. Then it's ectoderm, the nervous system is ectodermally derived, and the ectoderm in Drosophila is ventral. So this is an interesting difference that you'll see in vertebrates and invertebrates. It's dorsal in vertebrates. So, so does anyone, since we need to specify the neuroectoderm, it has to happen ventrally. Does anyone want to tell me how ventral tissue is specified in the developing Drosophila embryo? Do you remember from your lectures with Tina? So if you remember, you had to make you had to take the embryo, you had to specify anterior, you had to specify posterior, you also had to specify dorsal and ventral. So how did you guys specify, not you guys specify, but how did you learn that Drosophila embryo specified its dorsal ventral axis? Okay, Anjum.
Specifically on the ventral side, a toll ligand is produced that activates the toll receptor there, and then the ventral fate is kind of specific.
That's very good. Can you step one step back?
Yeah, I mean, there are a few genes that work. The bicoid, hunchback, all these genes are involved. There's a gene with G, I forgot the name. That
You're
actually.
right. Anjun, you're talking about anterior posterior patterning. I'm talking about dorsoventral. Can you remember? You're absolutely right about the anterior posterior patterning. Can you think back on the dorsoventral?
In the dorsal side, if I'm not wrong, pipe protein synthesis is restricted because of which.
Yeah. Okay. So let me bring this up. So I'm going all the way back to the maternally deposited factors that set up even before fertilization happens. So even if before it becomes an embryo, the mom pumps into the egg RNAs that already specify what anterior, posterior, and dorsoventral are. So the mom actually pumps in this RNA for this transcription factor dorsal. Do you guys remember that? And dorsal, so this is a picture of, I think this is actually showing you the protein, but if you can see over here, so we've taken the Drosophila embryo, sliced it, does like a salami, and we're looking at it like this. And what you can see is, so this is the dorsal end, and this is the ventral end. And fly names can be confusing because they're named after their mutant phenotype. Excuse me, mutant phenotypes. So the dorsal protein is called that despite being expressed in the ventral region, because in its absence, everything gets dorsalized. So here's dorsal in its expressed in the ventral region. And like any morphogen, you'll see much higher intensity of dorsal over here, and it kind of fades. And actually, if you were to look at the RNA, it kind of goes much more laterally as well. And what this does is, like any morphogen, it specifies it that downstream it turns on different genes at different thresholds. So right over here at the ventral region, dorsal turns on these genes, snail and twist. And do you guys remember what happens to snail and twist expressing tissues? They
ventralize.
Sorry, say that again, Yojit.
They ventralize the presence of.
Yeah, they do, but they also indicate mesodermal fate.
Oh, yes.
So when you get snail and twist, high levels of snail and twist, which occur at high levels of dorsal expression, you get mesodermal fate. And if you remember what happens to the mesoderm, through gastrulation, the mesoderm gets internalized. So that's over there, right? So can you see the? I wish I had a nice video. Maybe for next time I will do that. So you see how the mesoderm, which is snail and twist expressing tissue, is getting internalized. So now remember, we're showing it to you like this, and we're showing mesoderm coming in like this. But imagine in the whole tissue, it's kind of going in like that, right? Now, as a result, what happens is now we need to pay attention to this region, which is slightly lower levels of dorsal expression. That kind of, because of gastrulation, that kind of comes and becomes the ventral part, which is what you see over here as this blue stuff. And that is what is going to form the neuroectoderm. So it's ventral now, it's completely ventral because of gastrulation. And remember, the dorsal was slightly lower there. And at those concentrations, dorsal turns on short gastrulation. Okay, you can't see it in this image over here, but it turns on short gastrulation, which in vertebrates, maybe Bhavna will talk to you about this, is caudin, and keep that in mind. So now the entire ventral neuroectoderm is specified through an intermediate level of dorsal expression by expressing short gastrulation. Okay, so I really like this picture over here, which is showing you RNA. It's an in situ against 5 factors. And I love it because you can see this exact stage over here. You can see DPP. Does anyone know what DPP, the vertebrate homologue of DPP is? The flies love to give funky names to their genes. So DPP is decapentaplegic, and it's the BMP homologue. So you'll see in the dorsoventral axis when Bhavna talks about this, even though well, okay, wait, let me explain the figure first. So this is DPP in the dorsal domain. What it's not showing you is dorsal, because dorsal you've seen over here, but dorsal at high regions is specifying snail, mesodermal fate, and at slightly lower concentrations, it's specifying short gastrulation, which is ectodermal. And then here it's also specifying dorsal at those levels is also specifying these 2 factors, VND in blue over here and IND over there. And we'll come back to these genes later. But the important thing over here is SOG, which through gastrulation will become ventral over here. And this is your ventral neurectoderm. Okay, so just to put that in perspective of the whole embryo, so here's your early stage embryo. So this is stage 5. Did someone have a question? Is that why it blinked like that? Okay, no, okay, no. Okay, so at early stages of embryogenesis, you have those lateral domains in purple over here. You can see the neurectoderm, which is still, this is prior to gastrulation, and it's sitting on the kind of lateral side. After gastrulation, it gets ventral. And remember, at gastrulation, because the embryo is also growing in Drosophila, it's folding over onto itself over there. And then something special happens, which is the next stage that we will talk about. This neurectoderm remember I was saying it regionalizes the progenitor, and the progenitor kind of pops into the embryo. And that's the next step that we're going to talk about. So all of these, along the entire length of that neurectoderm, you're specifying these progenitors, restricting fate of the neurogenic tissue and allowing them to pop into the embryo, giving you something like this. And this is I suppose kind of analogous, not homologous, analogous to the process of neural tube formation that you will find in vertebrates. So it's not really a tube formation, but it ends up doing the same thing. So let's now, oh, I'm sorry. So yeah, there you go. So now we're in the business. We've specified neurogenic tissue. We now are in the business of trying to restrict it so that not everything becomes neurogenic. You're restricting it and trying to specify a progenitor. So do people have any questions so far? Okay, in that case, let's carry on and see how you restrict progenitor fate. Okay, so this is a really I keep having to refresh my memory about this because it's kind of and you'll see what I mean by that. Remember I told you that short gastrulation specifies neurectoderm, and now what that also does is it sets up the stage for proneural gene expression. Now, proneural genes are those genes that are responsible for neurogenic fate. And so what you need to do when you're restricting progenitor fate is you need to restrict the domain of expression of these proneural genes. And so if you look at it, so people have looked at what those proneural genes look like in that SOG domain. And here's an image of one particular one. This is an in situ showing, I don't remember. There are three proneural genes. There's AKET, SCUT, and lethal of SCUT that are involved in this early process. There are other proneural genes as well, but in this process, these are the three that play a role. And so in the image that you see over there, it's one of those three. I don't remember which one. Now, each one of those, so a few things I want you to notice. One is that, so we've kind of taken the embryo and turned it kind of ventrally so that we're looking at the ventral neurectoderm. So one thing, notice how we're going to call these proneural clusters because they're proneural domains expressed in a cluster of cells. So notice how they're like really neatly ordered, right? The rows and columns, very nice. So as a developmental biologist, you're thinking, how could an embryo bring that about? But file that away. You'll realize how it happens. But did someone have a question? Okay. So what's happening is that proneural expression gets clustered in this very ordered sort of way. And actually, each one of those clusters, like I said, consists of a small number of cells, a cluster of cells. So, and within that cluster of cells, what you want to do is, even there, not all of those will become neurogenic. Only one of them will. And that process of restricting from the whole tissue to this cluster, the cluster to the single cell, that is brought about by lateral inhibition. So we'll spend a little bit of time on this. So don't pay attention to the text over there. The gray tissue, let's just look at the colors. The gray cells in each one of those clusters, let's look at A to begin with, is your SOG expressing neurectoderm. So imagine instead of that, there's that whole tissue expressing this. And then you have those clusters of proneural expressing cells, which are the green clusters that I'm showing you. Now, superimpose this onto that whole embryo and imagine this, instead of those in the previous image where I showed you those, the in situs of lethal of SCUT and all of that, imagine these clusters organized along over there. Now, from within each one of these clusters, you want to select one guy to become the progenitor. And that also happens by proneural gene expression. But what you have to do is ensure that one cell amongst that cluster really shoots up its proneural gene expression and silences all of the others. And that one cell then becomes the neural stem cell, which in invertebrates is called neuroblast. So you might find me using neuroblast and neural stem cell interchangeably, but they mean the same thing. And then now once you've specified that neuroblast, some very interesting cell biology happens such that it pops out from that neurectoderm into the organism. And remember, I showed you that picture of that happening along the entire length of the developing embryo. So how do you get this, right? This is very interesting. How you get from a cluster of cells with proneural expression to one cell with proneural expression and becoming the neural stem cell. And the signaling system involved over there is notch signaling. So did you guys study all of the developmental pathways and notch in particular in your previous lectures?
We didn't go into the pathways.
You didn't go in, okay. So can you tell me what you learned about notch so that we can see what, I'm hoping someone will help me, because as I think IMT knows, I keep muddling up what happens in lateral inhibition. So I'll need your help. So can you tell me what you learned about notch signaling? I put a picture over there to help. Okay, if not, I don't want to put anyone in the spot. So if not, shall we just walk through it? And then if I'm getting something wrong, you have to tell me. Okay. So imagine in that cluster of cells. So now we can pay attention to the text over there. So we're now looking at the cluster that in A, which is expressing all of which are this light green color. And what I've written over there is that there's equal notch activity in all cells of the proneural cluster. So notch signaling, there's receptor ligand. Notch is it kind of works both ways, but okay, notch is the receptor and delta is the ligand, right? So delta can, if it activates notch signaling, then the receiving cell can activate the signaling pathway. So what happens in a proneural cluster normally is that the transcription translation of the receptor and ligand are kind of more or less the same. So every cell is signaling to each other. All cells have equal notch activity. But because transcription translation is kind of jittery process, right? There's some noise in transcription and translation. What will happen through that jitter is suddenly one cell will have a little bit more delta, just a little bit more delta than its neighbor cell. Now, what that does is suddenly that cell becomes capable of activating notch signaling in the neighboring cell. And that's what we're trying to show over here. So this cell, imagine, just got a little bit extra delta, and now it activates notch signaling over here. And that's this over here. So see how it's activating notch signaling. At the end of notch activation, what it does is it activates this complex called enhancer of split, which shuts off your proneural genes. So your A keeps cute, remember we were talking about that? So it shuts off your proneural gene expression. And the whole pathway is linked up in such a neat way that the proneural gene expression actually regulates the delta expression. And when that happens now, this signal receiving cell kind of, its fate is sealed. So it can no more become, because it's receiving signal, it's not even making the ligand anymore. And so now its fate of, by that chance event of getting signal is sealing its fate. It can make no more ligand. And so that tiny difference gets amplified. And now what in turn happens is that while this now cannot receive notch signal, this cell that is sending notch signaling to its neighbor, it cannot receive notch signaling anymore. And so what happens is the opposite over here, where enhancer of split now cannot inhibit a proneural expression. And so now it can make much more delta. So you see how this signaling pathway is taking a tiny difference that arises out of just jitter, noise in biology, and uses that to seal cell fate. I really am amazed at how at that, right? That noise is being used as something that seals fate. So is that clear? Is that clear what's happening? So now you've got this one cell where you have lots and lots of proneural expression because it became the signaling cell and shut off its neighbors. So remember, we're looking at one neighbor now, but it would have neighbors everywhere. So you can see how So you can see how now from having equal notch activity, this one cell inhibited all of the other cells from expressing proneural genes by activating notch signaling in all of them. And now once that is done, this cell can delaminate as a neural stem cell. I think I saw Atal put his hand up.
Yeah. So I was just confirming that this, I mean, the noise which is coming in transcription translation is just because of it's just a stochastic process, right?
Exactly that. It's just a stochastic process, and it's being exploited by notch signaling to seal the fate of such an important developmental fate, right? You're specifying a stem cell. The root
That's
of that
amazing.
is, yeah, exactly. I agree with you. It's very neat. Okay, anyone else with questions, comments? Okay, great. So I guess I passed my lateral inhibition class, huh? Okay, so at the end of all of that, we have, imagine this is, I showed you this one cluster, but remember this is happening along the entire length of the embryo, and you have neural stem cells popping up into the embryo. And this, remember I said, was akin to the neural tube forming. Okay, it's 10:26. I think we're doing okay for time. So what have we done so far? We're focusing on the invertebrates. Remember? Yes,
Just a question.
yes,
Sorry, yeah.
Utkarsh, go ahead.
Yeah. So how well is that delamination process understood? So
yeah, good question. You know, people have observed it. I don't think beyond the initial few descriptions of its occurrence, I don't think people have really looked at the mechanism. And I keep thinking that this is very interesting cell biology. So people have described it, you know, the cell kind of changes its shape, becomes more columnar, then it gets pinched at the bottom, and then it comes out. But I don't think people have studied it beyond that. I think this would be these kinds of things, you know, now with the way our ability to image, our ability to manipulate subcellularly, observe, you know, things like actin and myosin and all of these things with at the subcellular location. I think these are the kinds of questions that need to be revisited to really understand how these things occur. From getting proneural gene expression, how is this thing, this physical thing of being extruded out of a tissue happening? Yeah, that's a very interesting question. I don't think it's understood very well.
yeah, very cool to think about.
Yeah, exactly. Okay, so we've learned how you specify neurogenic tissue. We've learned from there how you restrict progenitor fate, and it delaminates into the embryo. So now, if you remember, we need to talk about patterning this. So remember, now we have neural stem cells along the entire length of the organism. Now we need to give it information that you are a neural stem cell at the anterior end, you are a neural stem cell at the posterior end, you are one that is much more ventral, you are one that is much more dorsal. So we need to give positional information in both these axes, dorsoventral and anterior posterior. So let's start with dorsoventral, because we kind of spoke about this already. So here's a picture that you have seen, where we had short gastrulation, intermediate levels of dorsal turning on short gastrulation, and I told you that those levels of dorsal also specify these other genes, BND and IND, and those are the ones that we need to think about now. So I'm sorry, this image is pixelated, but actually in the paper itself, this was the maximum resolution. So we'll have to bear with this. But this is basically the same thing that you've seen so far. So this is that early stage embryo. This is before gastrulation. So you have that dorsal over there. And this is the domain that we're interested in, right? That intermediate level of dorsal. And even this, actually, there are different levels. So this is kind of more dorsal. This is slightly less dorsal. And this is even less dorsal. And what people have shown very nicely is that even in these regions, you have differential gene activation going on. So at these levels of dorsal, we're going to ignore these things over here and jump straight to this end over here. A transcription factor called BND is specified. At these levels, a transcription factor IND is specified. And at this level, a transcription factor called MSH is specified. And I want you, again, you know, these are the Drosophila names. I'll tell you the oh, actually, maybe I should take that back. I think VN. So the vertebrate, is Bhavna still here, or has she escaped? At the moment I need her. But these are GBX, MSH, and NKX. I think VND is GBX, IND is NKX, and MSH is MSX or something like that. So pay attention to these in Bhavna's part of the course. You're going to encounter these very same factors. So remember, in vertebrates, it's the opposite. So the nervous system is dorsal. You know, our nervous systems, our spinal cords are dorsal, not ventral. We have a bellies ventrally. But despite that, the same genetic motifs you'll see encountering in dorsal-ventral patterning. So that's important to remember. Okay, yeah. So dorsal, the same transcription factor which was specifying neurectodermal pate is simultaneously specifying VND, IND, demarcating that, regionalizing that ectoderm into 3 distinct domains specified by VND, IND, and MSH. And VND, IND, and MSH also have regulatory interactions amongst themselves. So VND ensures that no IND expression can occur more ventrally. So it inhibits IND expression. IND does the same to MSH. So what is set up by the morphogen is being again refined and locked into very distinct, precise gene expression domains, just as you saw in the anterior-posterior patterning. So now what this ends up doing, so now this is a schematic to show you after that gastrulation what would happen. So again, we're doing that salami view of the embryo where this is one half and that's another half. So you have one stripe of VND expression right in the middle, followed by 2 stripes of IND expression laterally, and then the lateral most is MSH. And remember, this is going to be running along the entire, this is a salami, right? But if you were to turn it around like that, you have VND stripe like this, you have IND stripe like this, and you have an MSH stripe like so. Okay, so that's dorsoventral patterning. What does it do? You should be asking me that now, and I will tell you to hold that thought. We'll come to what dorsoventral patterning actually does. All you've done right now is made it molecularly different. The lateral part is molecularly different from the medial part. That's all you've done so far. Okay, anterior-posterior patterning. Anjum, you were telling us about anterior-posterior patterning. Can you tell us a little bit more? So can you start from, maybe I'll put this over here. Oops, okay. Okay, maybe I forgot what was going on. So anterior-posterior patterning, you're defining it along the anterior-posterior axis, right? So here's 2 examples of anterior-posterior patterning genes. I think this is Pushitarazu and either odd skipped or even skipped, I don't remember. And you can see how, very nice distinct expression domains along the anterior-posterior axis of these two, just these 2. There are others as well. And I'm going to put this over here because this is like a summary of how anterior-posterior patterning happens all the way from your maternal effect gene, so the stuff that mom puts into the egg even before fertilization occurs, all the way down to this. So Anjum, do you want to tell us a little bit about, you named some factors, maybe we could pop it into, bin them into these categories. You were saying something about hunchback and Anjum, are you here? Okay, does anyone else want to fill us in just very briefly about what's going on over here with anterior-posterior patterning of the whole embryo? So this is the neat thing, right? In all of this that you've seen so far, the nervous system just seems to be piggybacked riding on whatever's happening in the rest of the embryo. The embryo has to be patterned, let that happen. I'm going to use exactly those same gene regulatory networks and utilize it for my development. So it's really that. If you know embryonic development, you know how it happens in the nervous system. Does anyone else want to fill in?
And can I go ahead?
Yes, please, Shiva.
So the maternal effect genes, that is bicoid and nanos, they specify the anterior and posterior
Exactly.
parts of the embryo. So bicoid actively moves towards the anterior region, while nanos is restricted to the posterior part of the embryo.
Okay, excellent. What happens after that?
So after that, you have the gap genes, which specify the embryo into 5 main segments.
Great. Can you name some of those gap genes?
Like
All of this will
Krappan,
come up later. That's why I'm asking for, just so that everyone's memories are refreshed. Do you remember?
like Krappan and NURPS.
Yeah, Krupp, Knirps, Hunchback was one of them.
Yes.
Okay, that's a good beginning. So we made, you went from these two anterior posterior to about 5 domains using these gap genes, and then from there you refined it with pair rule genes, pair rule genes, gene
even
names.
skipped, odd skipped, Fushitarazu.
Fushitarazu, even skipped, odd skipped, engraved. Oh no, engraved is not. Okay, so pair rule genes, and after that,
the segment polarity genes, where each segment is conferred an anterior and a posterior region.
Exactly. Now, nervous system development uses these exact same genes to pattern it in the anterior posterior axis, and that's why I kind of poked you guys to give me the names, because you'll see exactly the same things coming up in nervous system development. So here's engrailed. So engrailed is the segment polarity gene, and we're looking at the ventral domain, and we're looking at those nice types of engrailed expression. The nervous system is coming out from the neuroectoderm at exactly this time. So the neuroblast, neural stem cells, are being specified. So here's the neuroblasts being specified. Each one of these is a neural stem cell that is being formed on that neuroectoderm and is ready to delaminate from there. But if you now were to superimpose these anterior posterior patterning genes onto these neural stem cell genes, then what you'll see is something like this. So what you can immediately see is that the neural stem cells that come out of this domain, this engrailed domain, are molecularly distinct from its neighbors. Right? Can you see that? Oops, sorry, I thought I had something else there. So this is engrailed, but now imagine all of those genes that we spoke about: Fushitarazu, odd skipped, even skipped, gooseberry, wingless, all of these genes that are involved in anterior posterior patterning of the embryo and the segments are somehow will make each one of these neural stem cells distinct from one another. So now what I'm doing is I've drawn these stripes, I've schematized all of that we just discussed. So here's your engrailed stripe. I've tried to align it with this blue stripe of engrailed that we have over here. I've drawn, sorry, my text is not visible. I've drawn a slightly anterior stripe of gooseberry expression over here. And here are your dorsoventral patterning genes. Here's your VND, which is more medial. Here's your IND, which is more intermediate. And then here's your MSH, more lateral. So imagine now, so over here, I've just shown 2 anterior posterior, but you can imagine many more of them, right? So what's happening is that as neural stem cells are forming, they're forming at the intersections of these anterior posterior and dorsoventral patterning genes. So you can see this guy over here is forming at the intersection of your engrailed domains that I've drawn over here and MSH, the lateral most. So it's molecularly different from this neural stem cell over here, which is not coming out from engrailed. And I'll tell you that the expression over here is of gooseberry. It's coming out from the gooseberry domain, but it's also coming out of the MSH domain. So it's molecularly distinct from this guy. And 3,5 is molecularly distinct from this one. And 5,3 is molecularly distinct from 5,6 because it's coming out at the intersection of IND and gooseberry, and not MSH and gooseberry as 5,6 is. So basically what I'm saying is all of this patterning event, anterior posterior dorsoventral patterning, what it's doing is making each neural stem cell molecularly distinct from one another. Am I making sense? Because this is really important.
Yes.
Yes. Okay. One yes. Thank you, one person. Are there? Is it generally? Shall I take that as a representative yes from everyone?
Yes.
Yes.
Okay, two makes a crowd. Okay, so what I'm showing you here is one hemi-segment, and I say that because each segment, remember these are segmental boundaries. So this is the posterior of one segment, and this is the posterior of the other segment. So within that is one segment, and this here is the midline, right? Because there's V and D over here. So on the other side would be a mirror image of this, and then of course each segment would have this happening, right? Because anterior posterior patterning, all of that. So what we've done is we've taken neural stem cells and we've given them molecularly distinct identities. What does this mean? What is my next slide? I've forgotten. So let's focus on one of those hemisegments. And you saw this picture, I've just enlarged it over here. And now we can talk about what does it mean to give a neural stem cell a unique molecular identity? Can anyone guess? If you have a unique molecular identity, it allows you to do something different, right? So what we know, sorry, that's just to lay out the axes for you. You're familiar with it now. That's the dorsoventral, and then that's the anterior posterior axis. Remember, we're talking about just one hemisegment over here. But what it allows you to do is something different. So here's a neural stem cell, which we're not looking at it at the level of the stem cell. We've waited for some time until it's made some neurons, and we're looking at what those neurons look like. And what you can see is these neurons are what the neuroblast 5-6 make, which comes out at the intersection of gooseberry and MSH. But if you look at its neighbor, that 7-4 over there, which comes out at the intersection of engrailed and MSH, it makes a totally different lineage. Now you can imagine this happening to all of them because they're all molecularly distinct, right? And here are all of those lineages, different types, and I'm showing you only, so this is, I'm sorry, I forgot to put the reference. This is from Lassin et al. 2016. And you can see these are all the neuroblasts, and you can see how this one is very different from this one, is very different from this one, is very different from this one, very different from, so basically each one of those molecular identities allows a neural stem cell to make different neurons. That is why you have to give spatial patterning. You're imparting the ability to diversify the type of progeny you can give rise to. Okay, so spatial patterning diversifies neuroblast lineage output. Okay, and there's a final thing about anterior posterior patterning that we haven't yet discussed. But if you remember this image that I showed you, on top of all of this anterior posterior patterning were the homeotic genes, the Hox genes. And what that means basically is, for the nervous system, is that, so you start off at the beginning, at the most anterior end, you have these homeotic genes called OTD and OTX. There's also EMS and EMX. More posterior to that are these genes called the PAX genes. You may be familiar with them. And then in the ventralmost region, that is where you have the ventral nerve cord or spinal cord invertebrates, you have the Hox genes. So on top of all of that we discussed with respect to anterior posterior patterning so far, which was segmentally repeated, on top of that, you now have an overarching anterior posterior axis information. So this is what we discussed, segmentally repeating anterior posterior patterning. Now what I'm telling you is, on top of all of this is the Hox identity. So right at the anterior end over here, you would have your OTD, OTX, you would have over here your EMS, EMX, you would have PAX. You would have your PAX genes over here, and then you start off in the ventralmost region, you would have your Hox gene identities over here. And what that does is, even though you're making, so if you pay attention, you have 5-6 over here, you have 5-6 over here, you have 5-6 over here. Ideally, all of these 5-6s, because they all come from the intersection of gooseberry and MSH, should be making the same lineage, right? But they don't, and they don't because they receive this extra information about anterior posterior axis imparted by Hox gene identity. And what that does then is gives each 5-6 a slightly different segment specific identity. So for example, this, just to orient you, this, don't pay attention to this. This is the outline of the nervous system in the very early larva or the late embryo. And these clusters of cells that you see are neuroblast 5-6. And these three clusters are neuroblast 5-6 in the thoracic segments. And these are neuroblast 5-6 in the abdominal segments. So you see how the same neural stem cell identity in more anterior domains are doing something slightly different from the, well, over here it looks like vastly different from the abdominal domains. And just to highlight that for you, those are the two neuroblast 5-6s from the two hemisegments of a thoracic domain, the T3 domain, and these are two examples from an abdominal domain. So the Hox genes are taking anterior posterior positional information and modifying the output of the segmentally repeated anterior posterior patterning. Okay, does anyone have any questions about that? Okay, so in that case, this is what I had planned for this lecture. So just to summarize, we learned how you specify a neurogenic region. We learned how within that neurogenic region, you restrict progenitor fate, specify progenitor fate, and how that then delaminates into the embryo. And then you take that and you provide spatial information to it, positional information to it, so that each neural stem cell can now generate different lineage identities. And what we'll talk about next week is how do you now ensure, so I mean, you could do a back of the envelope calculation and say, okay, how each hemisegment has these many neural stem cells, there are so many segments, that doesn't give me the 200,000 neurons that we spoke of. So how do you now get to those 200,000 neurons? And so what the organism employs is just some very clever cell division tricks to ensure that you get all of that. And of course, you're dividing over a period of time. So we'll talk a little bit about those cell division patterns. And then we will talk about temporal patterning, which takes within lineages. So I told you that you get these different lineages, but even within those lineages, there are different neurons, slightly different neurons from one another. So temporal patterning allows stem cells to take their spatial information and yet give out slightly different neuronal outputs. And that would be the end of today's class and a summary of what we'll do next time. So I guess we've ended earlier than I expected. Are there any questions? No? Okay, in that case, everyone's free to go, I guess. What's the system, Varsha?
No, so everyone just leaves the class.
Okay. So
leave the meeting, yeah.
Okay, great. So I put my email address there. If something strikes you after this, then please feel free to bug me afterwards. Okay, thank you all, everyone.
Thanks, Sonia.
See you on Wednesday.
Bye.
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 Thank
you, Sonia.
Thank you. Bye.
Thank you.
Yeah, Sonia?